Hello, I'm Ted Velasquez, and I will be presenting about single-domain antibodies, an innovation in monoclonal antibodies. Antibodies are also known as immunoglobulins. They are relatively large proteins produced in B cells. They have multiple protein chains. Each protein chain is coded by a different gene, and antibodies work by binding to epitopes in antigen. Each antibody is specific to certain epitopes on antigens. An antigen with many possible epitopes or specific sites could have many antibodies that bind with. The key here is specificity, and the specificity is why antibodies are useful in therapies. Specificity comes from the variable or V chains. The constant regions are generally biological in function. Antibodies never kill or destroy the antigen. They just target it for destruction through multiple means, mainly ADCC, or the cellular immunity, and CDC, or humoral immunity. One question now is, how do antibodies become specific? Well, it's a long process which begins with antibody variable chain genes in the B cells. The genes recombine randomly to generate diversity. The antibody is first expressed on a mature B cell, then B cells, which bind to antigens even somewhat, are selected. The selected cells then multiply to form the antibody secreting cells. This process is like evolution, but on a molecular level. Currently, antibody-based therapy agents include agents like trastuzumab, which is seen in the small image above, and it is used for treating aggressive forms of breast cancer. The MAB at the end means monoclonal antibody, and they are FDA approved like all drugs. There are other approved MAB drugs, the most recent one for lymphoma treatment with US approval in 2013. It is obinutuzumab from Genentech and Roche. Genentech was also the company behind trastuzumab and also insulin produced using transgenic bacteria. However, even with the specificity, the MABs are limited by the relatively large size and requirement for intravenous delivery, which is of course related to their size and structure. The very high cost of MAB treatments is also a problem, along with complicated production using mouse cells, shown in the large photo to the right. The single-domain monoclonal antibodies are small because they are based only on the variable heavy regions of camelid or shark antibodies, which lack light variable chains. However, they still retain much of the specificity due to the fact that the antibodies of camelids and sharks only use variable heavy regions for specificity. Their smaller nature means that they can be incorporated into new drugs, possibly those that need intravenous delivery. They may also be produced in prokaryotic cells or bacteria, as the structure of the protein is simpler and a single gene codes for the protein, unlike normal immunoglobulins, which have multiple segments and more complicated processing. The simplicity also allows for faster development due to smaller size and easier manufacturing. Companies researching single-domain monoclonal antibodies include Ablynx, which is a pioneer in the field. They have single-domain monoclonal antibodies derived from camelid antibodies. Haptogen, which is owned by YS, has single-domain monoclonal antibodies derived from shark antibodies. Aran Ordomantis, which is now owned by GlaxoSmithKline, uses human-based single-domain monoclonal antibodies. All these products are in the research and development stage. The large image on the right is sourced from the website of Ablynx, along with the graph on the left that shows progress of the drugs. One is near the end of phase 2 trials, and one will be going to phase 3 trials. One patent shown here is authored by Muruganandam et al. And was filed by the National Research Council of Canada. The patent was filed on May 25, 2001. Another one shown here is owned by Novartis, and it describes a monoclonal antibody with a C-terminal modification. It is more recent but has no photos and was filed September 13, 2013. Ablynx also has a patent which was filed July 16, 2010. And these are just some patents related to single-domain antibodies. The important one here is the one filed relating to a C-terminal modification due to the potential for innovation. Potential new ideas include pill-based delivery systems. New drugs based on single-domain monoclonal antibodies could change the way monoclonal antibody therapy is done. The smaller nature of single-domain monoclonal antibody drugs could mean that they could penetrate formerly unreachable areas like the brain. This is an example of improved drug delivery. Also, cells expressing single-domain monoclonal antibodies on their surface have been done, and an initial trial has shown that it works. So innovation could also include the kind of biotherapy which makes cells like bacteria attack the targets in ways like phagocytosis or other ways. This could lead to a kind of GM cell therapy. And polyclonal antibody can be done more easily since the structure of the single-domain monoclonal antibodies are simpler. Also, using the single-domain antibodies in applications where there is a need for specific binding, like using single-domain monoclonal antibodies for ELISA tests, or the example of the lab-on fiber sensors where it could benefit by attaching the single-domain monoclonal antibodies to the module and using the single-domain monoclonal antibodies' specific nature to bind only certain compounds for analysis. Since molecules can be engineered in many ways, possibilities are really endless when you consider the specific binding of a single-domain monoclonal antibodies and the fact that they can be attached to other components.